Glucose is our body's main sugar, but how exactly does it provide energy? Today, we are diving into this process, the process of breaking down glucose to provide energy, also known as glycolysis. So, glycolysis is a process that consists of 10 chemical reactions, and they occur inside the cytosol of our cell. Glycolysis consists of two separate phases, the investment phase and the payoff phase. The investment phase begins when glucose is taken into the cell. It is then converted by an enzyme called hexokinase into glucose 6-phosphate using a molecule of ATP in the process. After that, glucose 6-phosphate is further converted into fructose 6-phosphate using an enzyme called phosphoglucose isomerase. Then fructose 6-phosphate is then converted into fructose 1,6-bisphosphate by an enzyme called phosphofructokinase 1 using another molecule of ATP. Then aldolase will then split the molecules of fructose 1,6-bisphosphate into two molecules. One is dihydroxyacetone phosphate and another molecule is glyceraldehyde 3-phosphate. At the same time, dihydroxyacetone phosphate can also be converted into another molecule of glyceraldehyde 3-phosphate by an enzyme called triose phosphate isomerase. So, by the end of the entire investment phase, two molecules of ATP is used and two molecules of glyceraldehyde 3-phosphate is produced. Let's proceed to the payoff phase. The payoff phase begins when the enzyme glyceraldehyde 3-phosphate dehydrogenase converts the molecule glyceraldehyde 3-phosphate into a molecule called 1,3-bisphosphoglycerate, producing another molecule of NADH. But since in the investment phase we have produced two molecules of glyceraldehyde 3-phosphate, then two molecules of NADH is also produced. After that, the molecule 1,3-bisphosphoglycerate will then be converted into 3-phosphoglycerate using the enzyme called phosphoglycerate kinase, producing two molecules of ATP. After that, 3-phosphoglycerate will then rearranges itself into 2-phosphoglycerate using an enzyme called PGM or phosphoglycerate mutase. Enolase will then convert 2-phosphoglycerate into phosphoenolpyruvate, producing a molecule of H2O or water. Finally, phosphoenolpyruvate will then be converted into pyruvate, producing two molecules of ATP and two molecules of pyruvate. So by the end of the payoff phase, 4 molecules of ATP and 2 molecules of pyruvate and 2 molecules of NADH is produced. And if we were to sum up both the payoff phase and the investment phase, if we were to add them together, we can summarize that the entire process of glycolysis can provide 2 molecules of ATP, 2 molecules of NADH, and 2 molecules of pyruvate per 1 glucose molecule. So after learning the entire process of glycolysis, let's all proceed to conditions in which glycolysis can take place under. Glycolysis is a process that can take place under both aerobic and anaerobic condition. In aerobic condition, or when oxygen is available, glycolysis acts more as the initial pathway in order to produce pyruvate, a substance that is very crucial for the process of cellular respiration, which can only occur in the presence of oxygen. So in order to participate in this process, pyruvate has to first move into the mitochondria, where it will then be converted into acetyl-CoA, which further enter the citric acid cycle to produce what we call electron carriers, such as NADH and FADH2. After that, those electron carriers will then enter the electron transport chain, which further enter the oxidative phosphorylation to produce about 32 ATP. On the other hand, if oxygen is not available, pyruvate will stay in the cytoplasm. It will then participate in lactic acid fermentation in order to produce lactate. Glycolysis is most dominant in conditions such as conditions when oxygen is limited, example, vigorous exercises. Glycolysis is the only pathway that can produce energy without the need of oxygen. So in conditions like vigorous exercises, when oxygen is limited because they cannot be sufficiently delivered to different cells or tissues to support demand, glycolysis is the process that we use to provide energy in order to support life demands. Cells that lack mitochondria, like erythrocytes, also has dominant glycolysis. This is because mitochondria is a site in which many processes, including oxidative phosphorylation as well as the citric acid cycle, occurs. So without mitochondria, none of these processes can occur and none of these processes can provide energy. So cells with this condition tend to prefer glycolysis as its energy source. And that goes the same for cells with few mitochondria such as neutrophils, endothelial cells, and the islands. Last, we have proliferative cells such as lymphocyte T cells. So, recent studies have found out that proliferative cells such as lymphocyte T cells have high rate of glycolysis. This may be because glycolytic intermediates, which have been produced throughout the entire process of glycolysis, are crucial precursors for the synthesis of many macromolecules, including amino acids and lipids, which are needed in the process of cell division and cell proliferation. So let's all proceed to the clinical implications that is correlated with the process of glycolysis. The first one is what we call the Warburg effect. This is a phenomenon that occurs when cancer cells display high rate of glycolysis, even when oxygen is available. This altered metabolism is usually done to enable ATP production quickly and to support tumor growth and increase lactate production. This phenomenon has also been linked with many other conditions such as lactic acidosis and lymphoma. It should also be noted that while the Warburg effect is mostly found in cancer cell, it can also be found in normal proliferative cells such as T cell in order to support biosynthesis. The next condition is called hemolytic anemia. Hemolytic anemia is a condition where red blood cells are destroyed more quickly than the bone marrow can produce them due to deficiency in glycolytic enzymes such as pyruvate kinase. So, when pyruvate kinase are reduced, the process of glycolysis is also reduced, and when there is reduced glycolysis rate, ATP production is also reduced. When there's no ATP, the membrane's ion pump will stop working, and this will lead to reduced membrane integrity, which results in immature breakdown of red blood cell. Next, we have neurodegenerative disorders. Disrupted glycolysis can also be commonly found in neurodegenerative disease such as Alzheimer's disease, Parkinson's disease, and amyotrophic lateral sclerosis. Brain examinations on patients with Alzheimer's disease shows decreased glucose uptake and decreased aerobic glycolysis in regions such as the temporal and parietal regions. This condition is often linked with the reduction in glucose transporters such as glucose transporter 1 and glucose transporter 3, which prevents neurons from receiving enough energy, leading to mental decline, a very important key symptom of Alzheimer's disease. In addition, some other studies have also suggested that patients with Parkinson's disease and amyotrophic lateral sclerosis also exhibit compensatory increases in glycolysis of peripheral tissues due to dysfunctional mitochondria. So in summary, glycolysis is a process that consists of 10 chemical reactions that occur in the cytosol of a cell. They consist of two separate phases, the investment phase and the payoff phase. The investment phase uses 2 ATP, the payoff phase generates 4 ATP, 2 NADH, and 2 pyruvate. So summing up these two processes, the entire process of glycolysis produces about 2 ATP, 2 NADH, and 2 pyruvate. Glycolysis can take place under both aerobic and anaerobic condition. In aerobic condition, pyruvate moves to mitochondria to participate in the citric acid cycle, which further supports oxidative phosphorylation to produce about 32 ATP. In anaerobic condition, pyruvate stays in the cytoplasm. They participate in lactic acid fermentation to produce lactate. Glycolysis is most dominant in anaerobic conditions, cells without or with limited mitochondria, and proliferative cells such as T cells. Clinical implications of glycolysis include the Warburg effect, the phenomenon that occurs when cancer cells display large amounts of glycolysis even when oxygen is available. Hemolytic anemia, a condition when red blood cells break down more quickly than bone marrow can produce them due to deficiency in pyruvate kinase, a glycolytic enzyme. And neurodegenerative disorders such as Alzheimer's disease, Parkinson's disease, and amyotrophic lateral sclerosis, in which glycolysis disruption occurs. So that's it for today. Thank you for listening.